Looks like we have all of today's presenters here. So I think we are ready to start. So I'm going to share. Can you see my slides?
Yes.
Great. So welcome to the Autumn 2021 Lifeplan webinar. So this webinar is going to be 1 hour long, and we are recording, and the recording will be posted on our website. And why we wanted to have this webinar now is because many of you have already sent or are sending all these Malay samples and soil samples and cyclone samples that you've worked so hard for. And we thought, as we are CBG in Canada is now starting to sequence the samples, we thought you would like to see what that looks like. So we asked CBG to show us a little bit of their process and what is actually going to happen with them. So that was the motivation for having today's seminar. And the program that you've seen, I'm going to first tell you a quick overview of where we are now with Lifeplan and what are the next steps. And then I'm going to hand it over to CBG in Canada to tell you what happens to your Malay samples when they arrive there. Then Petteri Lehikoinen is going to give a quick introduction to the Bird Sounds Global web portal that has just opened. We're going to have a quick prize giving to the most helpful team. And at the end, we'll have a question and answer session, which also is going to chair. And we'd like you to put your questions in the chat. And I think, looks like we have 75 people now. I think it's best if we mostly keep our cameras off throughout the webinar so that we don't accidentally crash Zoom. And we'll take questions in the chat at the end. So where are we now? The first year of Lifeplan sampling is coming to an end, and the global sites are getting ready to switch locations between natural and urban at the end of the new year. And as in all experiments, there have been a lot of surprises, but despite all the technical difficulties and lockdowns and permit problems and natural disasters, we have managed to collect a really impressive amount of data together. Our goal for what remains of 2021 is to tidy up the first year of data and metadata. So hopefully next week, if you've started sampling, you should receive a questionnaire about this first location that you've been sampling in 2021. We'll want to know things like habitat category, elevation. We want to confirm the exact locations of your traps and check which direction your cameras were pointing, things like that. And we'll also be checking in about any apparently missing data, just to make sure that we definitely have everything that we are supposed to have from you. And our goal for next year, 2022, is to put together a full year of data. So we want to have all the teams sampling by the new year, if at all possible. Where are we with this now? We have 58 teams sampling now, which is the green dots on the map and a lot of the red dots as well. The red dots mostly are missing some contract, and some of them are already sampling. But do remember, please, that we can't actually use your data if we don't have the contracts in place. So in terms of data amounts, we'll go over these numbers. We've collected some 2.4 million images, some 8 million minutes of audio, almost 3,500 cyclone samples, 1,352 soil samples, and Malay samples, which you'll be hearing more about. We've collected about 1,821 in this first year. So what's the next step? What are we doing with your data? Our next major goal is to be able to tell you what species were in each of your samples. And there are many steps towards achieving this goal. But for all of the data types, basically, we first need to isolate the things that we are actually trying to identify. We need to build some kind of reference library of species corresponding to whatever the segment is, and then combine those in a species identification step. With image data, we are at the sort of isolating animal step, where we are removing humans and empty frames to be able to extract just the animal frames for identification. Today we will be hearing about where we are with audio data. So here we're already able to isolate the animals, but we are starting to build the reference library for species identifications. And Bette will tell us a little bit more about that later on. But mainly today we are going to hear about how to turn malaise bottles and spore samples and soil samples into these barcode DNA sequences, which we will then be identifying as much as we can. So that's it from me for the introduction, and I'm going to hand it over to CBG in Canada, and I think Yevgeny is here.
I am. Thank you, Beth. So I'll probably turn off my camera when I'm presenting, but I need to share my screen for the slides. Can you see that now?
Yes, that's great.
All right, wonderful. So I probably had too much caffeine last night, and I decided to have a little fun with the title for the presentation, but I think it does convey the message. We definitely deal with a lot of material at this facility, but before we actually go into the workflow, I would like to do a little introduction for our DNA barcoding facility. Just in case somebody who is joining us today hasn't been aware of the amazing capacity that we have here, and it's a over 50,000 square feet research facility with a couple of buildings that were specifically constructed to support the massive undertakings of the International Barcode of Life community. In 2007, we launched the CCDB Labs, the biodiversity building, which is currently is a home for our Canadian Center for DNA Barcoding, as well as some spaces occupied by Limnatron. A few years later, the Center for Research Genomics came in, and it's a new home for CBG collections, our expanded DNA archive, more advanced data center, as well as various administration and plant collection. And there is an old facility that had many uses. We used to run training programs out of it. This was a place for students and postdocs. We even tried to do COVID diagnostic at the beginning of the pandemic there. But currently we are repurposing as expansion to our bulk sample prep and storage space. And we definitely serve for the international barcode community with more than 50 countries sending samples to us directly, and that represents over 200 research organizations in various sectors from government to academic and private entities. We have processed more than 9 million samples through a single specimen workflow. It's hard to even estimate how many millions of samples went through the bulk specimen workflow. And at the moment, about 75% of all the reference barcode records in bulk have been generated by the facility in Guelph. We do have analytical capacity that is unrivaled, and we can support large-scale programs. We also invest quite seriously in DNA preservation so that we can actually use that resource for other research when permitted by biological material transfer agreements. But all that space and equipment doesn't really get the work done. So I would like to give a shout out to a few key people who are heavily involved in Lifeplan sample processing. So it helps to put the name to the face and multiple stages that the sample goes through. So some of the faces you may be familiar. We had some new team members joining us recently. So we'll save on the introductions, but please know that it's not just a few people, it's an army of people who are dealing with the samples when they arrive to Guelph. And the samples do flow from all over the world, either they aggregated in your local hubs or they come in directly to Guelph. We are quite accustomed to see large shipments coming into the facility daily. And I believe this is up-to-date count for how many Lifeplan samples have been received. But we can verify this at the end of these questions. So when the Malaise sample arrives to Guelph, we do have a couple of options, generally speaking. There is an opportunity to take all this multiple individuals, sort them out, and send through the individual single specimen workflow. And it definitely has numerous advantages. You can do species discovery, identification, you maintain the link between the produced barcode sequence with the voucher. You can get abundance data when you analyze a lot of specimens from a particular habitat, in addition to the occurrence data, but it can be slow and it does come at a high cost. Clearly, the road to hell is paved with good intentions, and so is the road to metabarcoding, but it's a compromise. So in some cases, it can be quite justified to leverage the high processing capacity of the modern sequencing platforms, so you can save on the cost and you can quickly assess, or I'd say quicker, assess what's in your community. And there are various applications to metabarcoding. And there is definitely something in between, but we are not talking about that application. That's what third generation sequencing enables us to do. And we'll try to focus on this pass for the Malaise trap samples. And if we do have the time, we might quickly revert back to the single specimen workflow. So it's a complex multi-stage process where each bottle, when it comes into the facility, needs to go through the accessioning. We need to collect all the information that we have and put it in the database, and then start moving the samples through the various analytical protocols. We need to remove ethanol, we need to collect data about the weight, and get DNA extracted, amplify our libraries, prepare them for sequencing, and send them to a sequencing platform. We get the data, and then we do something exciting with it. So a little bit more about what happens when the samples arrive and what it might actually look like. A single 24 cubic feet chest freezer actually holds 360 of those Malaise trap bottles. And I'll explain a little bit more later where that number comes from, but it's just enough to fill one lane of another seq run. So to get enough material to fill that run, to make it the most affordable and efficient use of the sequencing real estate on the flow cell, it takes up about 2 freezers of the material. So clearly with that volume of samples coming in, it's always a constraint to have sufficient floor space to keep adding the freezers and to accommodate storage of this material. Clearly the information needs to be submitted to the database and the LIMS to enable tracking through the analytical steps. And once we initiate processing, we have to set up the vacuum manifold that we will be using for filtration of the ethanol from the samples. We decant it from the Malaise bottle over the filter. We secure insects on the filter by rinsing it from the filter. And then we need to move the samples back into the bottle, record the weight, add lysis buffer, and incubate on the shaker in the incubator. One key element of this particular workflow is there is no sample homogenization, so it's non-destructive lysis, and there are 2 reasons for that. One, because of the bulk imaging workflow requirements, that is executed after initial sample processing, as well as opportunity for those samples that already went through bulk lysis to be individually sorted and re-enter the single specimen barcoding workflow. We've done pilot studies to confirm that it's actually quite feasible, and we can get high success recovery from samples following repeated processing. So DNA extraction is a classic bind wash elute using membrane-based filter. We use a 96 well format for those plates, and it's a well-known glass fiber-based protocol that we've been using in this facility for over a decade. It's highly scalable and can be executed with very little modifications without heavy equipment. Mostly manually. It can be run as a semi-automated DAC method on one of our FX instruments, or can be fully automated by integration with Cytomat's DAC robotic transport, robotic arm, and a robotic centrifuge that can support 5000 G centrifugation. The protocol to extract DNA can be replaced with spray if further scale up is required, but we're not at the stage yet where that would be the primary constraints. Actually, handling individual bottles and doing lysis transfer is what's occupying a lot of time. The bulk imaging, I won't talk about it too much, but want to point out that it's a bit of a compromise. Generally, if you do imaging in the barcoding workflow, you do that before you damage the sample in any way, so you get the most accurate image of the voucher, but it becomes a bottleneck in processing, and it's more feasible to do that after. You also don't have to worry about cross-contamination between multiple samples when you do have to image, and you also not risking as much to lose the samples. But ultimately, it can be a step that can be executed after bulk lysis has already been completed, and the specimens might be slightly distorted or discolored because they have been exposed to harsh chemicals. As I mentioned, it requires a lot of capacity to store the tissue samples, the ethanol preserved material, the lysates, if we ever need to repeat DNA extraction or we want to reanalyze the material. So we have a substantial capacity to preserve samples at minus 80 degrees or minus 80, excuse me, or negative 20. With limited floor space capacity to expand. On top of it, with aging infrastructure and high cost to maintain, repair ultra-low temperature freezers or replace them after the end of life, we start and explore options for off-site storage. And some of the scenarios that we're now looking into is subscription model for off-site facilities where you may have to pay $1 per plate monthly. So it doesn't seem like a huge number, but if you look over the duration of the project, it quickly adds up, and it's definitely more costly than just buying equipment on site. Sample repatriation, shipping charges, that as well can add to cost for processing if it needs to be taken into account. It's just something that we need to be mindful, and that makes us to arrive to certain compromises. But let's get back to metabarcoding. There are certain challenges or complexities when you're dealing with the bulk samples. You have size variation, you have different abundance, overrepresented species. There is some rare organisms. On top of it, you're also dealing with the fact where specimens bring their dietary items into the bowl. So if you detect a species, it doesn't necessarily, it was present there as a voucher, it may be just remnants of someone's meal. But we have explored kind of how many rates you would ideally need to have, at least to have an adequate representation. So based on this study, we generally aiming for at least 1 million per sample. And mind you, in this particular example, it wasn't the most challenging scenario of all. There were only 375 species with single bean members represented. So we need to tackle those factors and keep that in mind when we're setting up our meta-barcoding experiments. The primary choice, I'm not going to talk about non-CO1 barcoding or alternate markers. We'll just focus on terrestrial reports in the malate traps and specifically the approved marker for animal barcoding, the saccharomycidase one. And this publication did a in-depth comparison for various options. So decision for this specific version of the workflow was to go with BF3, BR2 primers that give us just enough of the lengths to meet the maximum constraint of paired-end sequencing on Novaseq platform, and they perform quite well compared to a lot of alternative options. So again, that's a bit of a compromise. We also mindful of the fact that replicates are generally recommended. In multiple works, it's been demonstrated that it's beneficial. So this is a visualization of one of the plates on such of the runs that we are assembling from our bottles, where these 3 samples will be 3 different replicates from one sample, and you can fit up to 30 bottles on the plate, and each plate will include negative amplification and extraction controls. The indexing setup for these libraries is done with inline barcodes. Illumina sequencing generally plagued by issues with low complexity libraries. So by starting the sequencing, pretty much this will be our insert. By starting the sequencing at the area where the barcode sequence begins, helps to make heterogeneous first base calls when the clusters are called during the sequencing by synthesis on the flow cell. So the initial amplicon in PCR1 is generated with 418 base pair target with BF3, BR2 primers. We use the same priming sites for PCR2, just very long tails for P5 or P7 adapters at the end, without Illumina inline indices that would normally be in this sites. So this is our emit. And we also sequence half of the library in reverse direction by flipping those adapters that anneal the template to the anchors on the flow cell. The run itself ends up being an aggregate of multiple plates. Each well will have a unique combination of the indices. And we have a combination of 24 inline barcodes for in reverse. They vary in length from 6 to 10 bases. And when we need to demultiplex, we fill in the required bases from the oligo sequence for the total length of 10 nucleotides. This gives us 1,125 unique combinations in this set with 12 96-well plates. So what you see here at the top, there will be reverse barcode, primer with unique sequence, and intersection will be another unique version for forward primer. So each well in this 12 plates will have a uniquely labeled amplicon. So it takes 360 samples with 3 replicates on this setup that get pulled to fill a lane. As I mentioned, each well will have negative amplification, negative extraction controls. The last plate on this set up will have synthetic and non-mock community control, where every sample has been individually sequenced and verified its identity. So because a S prime flow cell can hold 2 libraries with 2 lanes, we can have up to 720 bottles on a flow cell. So this is where those 360 samples per lane or 720 bottles per flow cell come from. And we aiming for 1 million reads per sample. With this complex indexing setup, we have to rely on automation to ensure that the transfers are clean and that nothing accidentally gets mixed up, so we have full traceability. We have to label all plates with barcode scannable labels, human readable labels, triple check the pairings when we do the setups, and we also have to generate reagents in pre-made validated batches and clean environment with dispense of the master mix, addition of the forward and reverse unique primers to each of the version of the plates, and then transfer of the PCR 1 diluted template into the pre-made template-free PCR 2 mix. Once we have our applicants generated with 2-step PCRs, then we need to pull them and run the library to get it ready for sequencing on NovaSeq. For some applications like GSSP material, they go through different platforms with different markers, but this specifically talks about how to prepare the library for sequencing, sometimes not necessarily for the NovaSeq. So you have to do the cleanup, you have to do size selection, so you remove anything that's below your target range or anything that you have in the sample that is longer the required sequencing. Illumina doesn't like to have very long inserts present that can interfere with the run quality and can actually result in the run failure. So while a MacBit and PureBit cleanup and size selection usually cheap and fast, it's not always the case if you have to repeat it multiple times. So if you have opportunity to do size selection on BluePippin, it can be more precise, but it is a little bit more expensive and lengthier. You can use qubit fluorometer to do quantification to ensure that you do have sufficient library. You visualize it on tape station or by analyzer to verify that the library is the right size. You don't have anything too short or too long present in library, and then you're ready to send that for sequencing. So when it comes to NovaSeq, it has incredible throughput, but only if you sequence something like 75 or 150 bases. But if you want to get the maximum read lengths from the NovaSeq platform, you have to rely on S prime flow cell, and it has significantly lower throughput. Yet you still have an incredible amount of data that you can recover with over 1 million reads after merging and filtering, sometimes received per sample. So the reliance on data processing capacity is critical. And there are various ways to go about bioinformatic processing. You can have a custom data pipeline that can be written in Python. You can use the tools, but we mostly rely on Embrave that's our informatics team developed specifically to support the analysis of metabarcoding samples. But before we get to submit samples to Embrave, we first need to move, even in a compressed format, tens of gigs of data from those FASTQ files, because we use inline barcodes that generally never demultiplexed by an instrument, and you have to do custom demultiplexing and read margin, and at that point you can move it through the steps for quality filter and adapter removing, dereplicating your sequences, clustering them into use, and assigning them to taxonomy, which all of the steps that Embrave supports. The assignments of those taxonomy are only as good as your reference library, however. And anytime you start metabarcoding samples in a new exotic location where we haven't done a good job of building that reference library, you're going to run into a lot of problems. So this brings us back to the SQL single specimen workflow, where other than going straight from the bottle to individual sample sorting, you can actually enter those bulk samples that have already been processed and individually plate them, one sample per well, and run them through the robotic MACBIT based extraction. What you have here is a pin tool where you can actually see the brown bits on the immobilized on the cover plate that have been picked up from a source plate or about to be diluted into the DNA plate. This way, we can actually achieve serious increase in throughput and cost reduction because we don't use as much of robotic tips. So we can move thousands and thousands of samples through this workflow during the day. But other than that, the steps are quite similar. You lyse, you extract, you do PCR, you have to do index in PCR, but you do it in 384 format. And then you go through multi-stage SmartBell library prep workflow with sequencing on SQL, and followed by the analysis to assemble those sequences into the finished context with submission to Bolt. The reliance on automation in this high throughput workflows is paramount, and for a long time single specimen imaging was inconceivable or was a serious constraint to the workflow. But because of the automated imaging with Keynes microscopes, every individual in one of those wells on the plate now can get a shot and have its voucher image captured. It may not be a perfect image, but it's sufficient to do sample validation when we need to do sequence interpretation in the absence of taxonomy. It's almost like we are effectively enabling reverse workflow when we enter into this SQL-based barcoding without knowing much about the taxonomy for organism, except that it came from the malaise trap, and maybe we know the order of the organism just from quick assignment during sorting. It scans through the plates automatically and snaps a shot, and it can move through the plate in less than 30 minutes. Pretty amazing thing. Of course, and when we run SQL, we have to work with batches, very large batches of samples to actually make that workflow affordable. And the reagent manufacturing is not a trivial assignment as well. So each of this plate will end up having a PCR master mix with a well in 384 well plate with unique combination of primers, dual indexed. And we have to use automation to do extraction and PCR setup because it's almost impossible to expect high throughput processing of the samples in 384 format with manual pipetting. We do keep all the DNA extracts in a massive fleet of ultra low freezers, but we far exceeded that and as I mentioned, considering outsourcing that or finding additional space. Amplicon indexing with two-stage PCR would be similar to what we do for our NovaSeq libraries, just a different structure of the adapters, but the same principle. When you have the priming sites for your low-site specific primers, we use Cocktail from LabF and LabPar primers with Fulmer primers, forward and reverse. This was they are tailored with the universal plug-by adapters, and in the round 2 we use the universal adapter sequences to attach unique molecular identifiers to the amplicons. And we have 96 forward and 96 reverse combinations of those that we use for a single set of 96 well plates that results in combination of over 9,000 unique amplicons. But two-step PCR is also too complex and too expensive, so we merged it into a single step where we're going straight to generating the amplicon with dual indexing, and that definitely helps to reduce the cost. But we did run into a complexity where initial use of degenerate primers had to be replaced with primary cocktails to simplify data processing on mBrave and improve recovery of reads from demultiplexing. So we have an ability to run almost 9,000 samples on a single 1 million smart cell on Sequel 1. But we can do even more when we add a second layer of multiplexing. And instead of using just amplicon-based indexing, we can also use index smart bell adapters. So we can have virtual pools, if you will, of virtual smart cells on a single 8 million smart cell on Sequel 2. So we can suddenly multiplex tens of thousands of samples on a single run. But to get something like that is not necessarily trivial in terms of building that volume of samples. And if anything goes wrong, all of a sudden you have to repeat 2 months worth of work. And that's what Sequel 2 can ultimately deliver. With current capacity, it's going to take CCTV Lab up to 2 months to get enough DNA extractions and PCRs produced to fuel a run on Sequel 2 with 4 of those smart cells. But the technology has been improving through these years, and we've seen a steady growth of data outputs from Sequel platform. So when we started with barely 5 gigs of data from a single smart cell, this volume of data actually has exceeded 4-5 times that with the improved chemistry and increased movie times. So there is definitely a huge potential for this technology moving forward for single specimen barcoding. Once we have received the CCS data from our Sequel instruments, they go through the analysis on mBrave, and the resulting sequences end up in the Barcode of Life data systems where they can be analyzed and compared and validated. And I won't go into details because I probably don't have that much time, but I'll highlight the fact that since we moved to primarily using PacBio smart sequencing technology for single specimen barcoding, there's over 2 million, it's actually probably even more than this right now, it's a bit outdated slide. We have generated more than 2 million barcodes using smart sequencing technology. And I am always sensitive about forgetting somebody, and I want to remind that this amazing work is done by teams of people, and it takes a lot more crystals to get something to shine. So aside from the CCDB team that is doing all the hands-on work in the lab, we are supported by incredible teams in collections, informatics, and admin that keep this facility running. And there are some people who even left CBG, but they have immensely contributed to the success of this organization. So I would like to have them appear on this slide as well. Thank you. I think this is all I have as a brief overview.
Thank you, Evgeny. Thank you very much. Next in the program, I think we're going to go straight to Petteri Lehikoinen with the Bird Sounds Global web portal, so we can leave time for the question and answer session at the end. So Petteri, are you ready?
Yes, thank you very much, Wes, and I'll try to be quick with this one. I guess I don't have that much time either. Yeah, so I guess now you can see my screen.
Yeah, we see.
Yeah, perfect. So Yeah, I'm going to talk about the Bird Sounds Global web app, which we launched yesterday, but also more about the kind of citizen science and also identification of all the identification of bird sounds in general. And I'm going to start from the very scratch, which is the overarching aim of the Lifeplan to generate understanding of global biodiversity, and which the 4 kind of major steps to meet this aim is to gather data, process the data into a analyzable format, analyze the data, and then predict the results. And what we are here now, what best told that there's already enormous amount of data flowing in, and we have, like in the previous presentation and previously, we've heard that there's also a good process going on within all the kind of fields and processing all the data. But now we are focusing to the audio side, which will have to be done through machine learning. So we have to ask the computer to identify the species and found the species from the audio, because the data are so enormous that we can't listen them through by ear. And for this, we have started a pilot, launched a pilot already quite some while ago with the Finnish audio data. We have 2 million minutes of similar data, which Lifeplan is also collecting. And there's a kind of a citizen science web application to produce the training data, to which then is used for the machine learning part to identify the bird sounds. And in this, there's kind of 2 tasks for the user in the pilot study that we asked, or we first identified the kind of letters or templates for each species, which then search through the audio data, kind of similar sounds or sounds which are variously in similarity to that, and ask the users to identify if this is the same species as in the initial template. And in the second phase, we asked or provided the users a 10-second segment of audio data and asked them to identify all the species vocalizing in this segment. And with this, we kind of got this local pilot that this is the kind of pipeline for this. We made made ourselves the templates. Then we asked in this citizen science project to identify these similar sounds, but also the 10 recording sounds. And then the similar sounds then were used as the local training or training data for the local models to identify the Finnish birds. And partly also this 10 recordings were used for this, but mainly to kind of test these local training data. And with this kind of process, we have been able to produce kind of good local models to identify Finnish birds. But the problem here is now to expand this for a global scale, is to do the similar sound, but again, the kind of the magnitude is so much bigger that we have to use or go through a kind of shared effort, which the Bird Sounds Global now is the first step that we built. We'll build the global templates for all the bird species in the world, and then go through the similar steps. And we have to also launch another citizen science project for kind of then producing the training data and also the testing data for the global models with using the Lifeplan data, audio data in it. And the Bird Sounds Global, in a nutshell, is the users are asked to compile 10 representative sound features for each of the bird species in the world. And when everything has been done for the all 10,000 species, the end product will then be an open access sound feature database, which will be very, very valuable for the kind of further processes of the automated identification of bird sounds data. But also this will be published in a peer review journal, and all the users which have kind of given certain input to building up the templates will be invited as co-authors. And before we go to the Bird Sounds Global, I will briefly show you how it's working. I want to say that I'm merely a messenger here, and I want to acknowledge the kind of bird sound identification team, but also all of you who are kind of providing data for this and gathering data and so on. But Patrick, Panu, and Otso are mainly responsible of building up the models and methods for automated data identification, and Meri Rannisto from the Finnish Museum of Natural History has built up the web portals. And yeah, now we can go to the Bird Sounds Global itself, which has 3 kind of leaflets or tabs where you can go. First are the quite short introduction and also instructions for and then the main part is the list of bird species in the world, which here are all the bird species in the world, but you can filter them with the continent or the order. For instance, if we want to now focus on galliforms or the wildfowl of sorry, I was meant to put Asia here because Europe has mostly been done already. And then you can also filter or sort the list with the validation counts. You can also see only the species which has not been validated yet. And for instance, here I could then choose the chestnut-breasted partridge, which the template hasn't been done. So here below the templates, which are not yet defined, are the audio data. There's 5 audio data for each of them. They all have been already, an algorithm has searched for the kind of possible candidates for these templates. And you can listen through here and you can see that, okay, here that there's a target species. By double clicking this candidate, for instance, you can open it. Then we want to little bit reframe the templates so that there's only this kind of sound feature which is repeating in the system, and then we can confirm it and then repeat this step for 10 times, and the species templates are already made. I'm not going to go through all the 10 templates, but show you that then when somebody has done the 10 templates already, then they appear here simultaneously all, and you can listen them through here without even opening them. Just listen through and check everything is correct, and then confirm all the templates here. And then after 2 users have kind of confirmed the same templates, then the species is ready and we are very happy. And then there's also the results section, which shows how many species have been already validated. And here I want to say that we are extremely grateful for all the test users from the Lifeplan teams who gave their valuable comments to improve the system before we launched it. But also they have done a great job, or lots of work already. 10% of all the species have already been validated and templates built. So now we are at the stage that these kind of their improvements have been taken into account, and the portal is ready for advertisement, which we will start to advertise now. And we are also extremely grateful for all of you if you can pass on the word of the global or Bird Sounds Global to your networks and so on. But we will be also advertising it ourselves, of course. And if you have any questions about this or more of them, I'm really happy to help. Thanks. I guess this is all that I have to say.
Thank you, Petteri. I think we're going to save all the questions to the end, as we are running short on time. We're going to have a quick prize giving session. So I think Deirdre is here, and ready to do the prize giving.
Hi. Yeah, so very quickly, the first person to receive our fancy Lifeplan t-shirt and Lifeplan cap is someone that very early on, when we realized we were having a lot of issues with the micro SDs from the AudioMoth, contributed a lot by communicating with myself and with Tommy and troubleshooting their own AudioMoths and testing their micro SD cards and sending us results. And that person is Jorge from the team Baja California Sur in Mexico. So thank you very much, Jorge. I will be emailing you and we will be sending you one of our lovely t-shirts and caps. So thank you very much for your help.
Thank you, Dee. And thank you, Jorge, very much. Okay, that is the main program. And we are now ready to start the Q&A session. And Otso, Otso, I asked you to chair this. So if you can start looking through questions in the chat and maybe throwing them along to people who might be able to answer them.
Okay, thanks a lot. First, I would like to say that thanks a lot for the speakers today. That I think you had really great presentations. I was enjoying all of them and learning a lot. So I'm looking at the chat and I see that during the presentations there were quite many questions, but they actually have all been answered already. So just place your new questions to chat and we will answer them. Yes, there's a question to Petteri. Should we spread the voice about the portal to our colleagues to increase the number of participating specialists? Yes, I think Pepe already said this actually at the end of the presentation, that if you have colleagues who know birds and you think who would be willing to spend some effort with the portal, please invite them to take part. So this is of course open to all the Lifeplan teams, but it's also open to anyone else who knows birds and would like to contribute. Let me take from some of the earlier questions, actually from Ricardo was asking about, is it worth taking pictures of the malaise samples? I mean, this was before Evgeny presented that we do actually take bulk images of all the samples. So you're asking that whether it would make sense for you to take the pictures beforehand. And no, I'm not exactly sure if this is a good idea, because it may cause some contamination and all of that if you do some extra, moving around of the samples before you send them. Maybe Evgeny could comment on this, whether it would be problematic if the teams do take pictures before they are sent.
Jerry, you want to take this one?
Yeah, sure. We definitely don't want pictures taken ahead of time, and we really don't want the bottles reopened after they're taken off of the malaise traps. Contamination is a serious concern. We're using high throughput sequencing technologies that are so sensitive, and we don't want to waste any of our reads on human DNA or the sandwich you may have had at lunch. So
Jeremy, sorry to interrupt you, but there was just a clarification that they are just pictures from the top of the bottle without manipulation. So clearly this is not problematic. Of course, there's no problem of taking those pictures. But it's good to know for everybody that we are taking, or you guys in Canada are taking the pictures of all the samples, because we want to combine image-based and DNA-based identification of the specimens. And so you will also have, of course, in the end, access to those images yourself if you want to have them.
Yeah, that's right. And I think we will, I think tomorrow we're having a smaller meeting to discuss that transfer of the bulk images. And yeah, just to clarify, we will be taking those after metabarcoding has been completed. Again, even from our end, within a really clean facility, we still have to worry about contamination prior to metabarcoding.
Thank you, Jeremy. Then there's a question from Jessica about Nagoya guidelines. Maybe Bess, you could answer this. So we would like to send malaise samples soon, but there are still some things unclear to us in relation to the Nagoya guidelines. Is the recipient responsible for the required documents, and what documents do we need to fill out, and where do we get them?
Yeah, so Nagoya guidelines, they vary very much by country. So it depends entirely on what country you're sending from. But it will, so we have a Nagoya lawyer who has gone over all the countries that we're collecting samples from and is checking on what the relevant permits are. So if you do need a special permit, then you should know that by now. So it should have been part of your sort of MTA negotiations. Yeah, so usually if you have an MTA signed, and that means that we've checked through that there's nothing else required and it's just the MTA that is between us, and you don't need a special permit.
Okay, thanks, Bess. At the moment, there are no more questions. So if you have any questions, it's a good possibility to send a question. Okay, then there's a good question from Jorge to Petteri or to the bird identification part. Some of us have little experience with bird songs. Would it be an error to register and practice with the site? And when Petteri showed this Finnish pilot study, which generates like the training data for the Finnish birds, not the templates, but the training data. There we actually encourage anyone to use it. You don't need to be like a real expert in the birds to use it, but there's a questionnaire which kind of asks your self-evaluation about what is your level of expertise, so that then we can give more weight to those data points where people kind of think themselves that they are experts. And also we do ask several people to look at the same sound sample so that we can see how consistent the results are, and in this way we can also evaluate the quality of the identifications. So that for the main citizen science project, not the template building that we are talking about now, but the main generation of the training data, I think we would encourage anyone to take part. But now for this template building, we would only encourage those to take part who are then confident that they know the species, that they either know the species beforehand based on their own experience, or that they know they are able to use external resources to kind of learn the species, so that in the end they are confident that they actually are able to disentangle the focal species from some other species that may be singing in the background. So I would say that this part is mainly for kind of the experts.
I could take a question here from Ricardo Diaz Delgado about the soil sampling. So you don't need to count 8 weeks from the last sampling. The idea is that if you're in the Northern Hemisphere, then your sort of sampling period is the period when the ground is not frozen. And over that whole period, try to sort of roughly divide it into the number of sampling events that we have, and try to spread the sampling over the whole year. So you don't have to wait 8 weeks. You can start as soon as you're able to take soil samples.
Yes, yes. I think this question is specifically about when you move from urban to natural or natural to urban. That whenever in that new site the season is the right, then you can go. You can take the first sample. Exactly. Oh yeah, and then I missed one question by Laura, which is actually for Yevgeny. That are you considering to receive team members or students to help and learn how to analyze malaise samples at your facility? So does Kelf welcome this kind of trainees or visitors?
Well, we used to have a training program, but we don't do right now. Also, any travel or accepting visitors during ongoing pandemic is still a concern. So definitely not at this time, but we definitely expanding and have a few positions like research technicians. I believe there may be some postdoc positions open in the coming months as well. So there are opportunities, but not specifically as a visitor training program at this very time. Jerry, you have anything to add?
Not really. I think we're just transitioning into receiving visitors. We've had a couple of visitors from the US that visited our specimen archive last month, and I think there's talk of having a couple of visitors in the winter that are from outside of the US. So it's slowly happening, but I think it's maybe a little too early to start inviting visitors for training purposes, but we'll definitely revisit that over the next few months while everything starts to open up. And I think we're open to that idea.
Do we still run the online training program, or that's also been suspended? Have you
I
heard of
believe
it?
that has been suspended, but
Yeah, okay.
yeah, I can double check and maybe we can comment on it in a future seminar.
But it's always good to follow up by email and check out the website for any updates.
I see that we are already a couple of minutes over time, but let's still use a couple of minutes to answer the remaining questions. So there was a question about if we can get the list of the locations of Lifeplan, like the coordinates or a map or something like that. And Wes already promised to put that on Slack, so you will get that. Then Jeremy asked that whether there is a plan to create a similar citizen science reference library building for the animal images captured by the camera traps. Yes, that is a plan as well, that after we have built the machine learning models that just like Wes explained, extract the animals from throw away kind of the empty images and those with people, then the idea is to maybe not build ourselves, but utilize one of the existing web portals to generate training data, and ask all of you to contribute to that also for the camera trap images. I see now there are no more questions at the moment. Just greetings from various places. Thanks very much for those. So maybe it's time to wrap up this seminar. Once again, thanks very much for the speakers. It was really great to see, you guys in Canada who do all the sequencing, that to appreciate that it's a lot of work that is happening at this end to do this magic of getting the DNA sequences out of the samples. It's actually a big factory that you are running there to do that. And especially I would like to thank all the teams that you have, started and continued the sampling. Is like a really big commitment, and I think we will together produce really exciting science out of this. It's a long process, but we are quite far. You have gathered a lot of data. The data processing is quite far. It's not in the end, but the methods are developing at fast rate. And so I think we are gradually approaching the stages where we can actually go for the first kind of publications out of the Lifeplan studies, like first maybe the data papers, the reference library papers, the methodological papers, and hopefully as soon as possible also the biological papers, that what do we actually learn about nature by doing this amazing global study with all of you. Okay, so I think it's time to close this webinar, and you know, we will keep on posting the newsletters, and also for sure we will have these kind of webinars also in the future. Okay, thank you.
Thank you.
And bye to everybody.
Bye.
Thank you. Bye, everyone.
Bye again.